Dear students, in the last video we have gone through some of the cell organelles and the continued part of the cell organelles let us go through. So next cell organelle is the major cell organelle, which is mitochondria. So let us start with the mitochondria. Mitochondria is very important cell organelle. Why? Because it helps in the production of energy, and that energy will be in the form of ATPs. Hence, mitochondria present in the cell are commonly known as powerhouses of the cell. These are commonly known as powerhouses of cell. Why they are called as powerhouses of the cell? Because mitochondria would help in the production of energy in form of ATPs. Hence, they are called as powerhouses of the cell. So, this process, production of energy in form of ATP, that take place by cellular respiration. It is also called as aerobic respiration. Aerobic respiration means mitochondria perform aerobic respiration. By that they produce ATPs. Hence they are called as powerhouses of the cell. So whenever we go through the discovery of these powerhouses, Kolleker is the name of the scientist, the one who discovered these mitochondria. So, and he teased them from muscle cells. Muscle cells means he saw them, he extracted them from the muscle cells for the first time, and such kind of cell organelles, how they are, will go through the structure of these cell organelles. Whenever we go through the structure, their size of these cell organelles, that is mitochondria, it varies from cell to cell. It depends on the type of a cell in which these mitochondria are present. So the size depends on that one, right? And even shape of mitochondria. Shape of the mitochondria, it also differs. Commonly, it may be spherical in shape, or it may be rod shaped. Sometimes it is especially spiral shaped in sperm. In sperm, usually it is spiral shaped. So whenever we go through the structure of the human sperm, that moment we will see here. The structure of human sperm, and here in this part, middle piece, it is called as, it is a head part, and this is middle piece. In middle piece we will find this kind of a spiral shaped mitochondria over here, right? So such kind of mitochondria is present in human sperm. So that's why it's a shape that differs from one cell to another cell. And whenever we come to the structure of this spherical shaped mitochondria, I am going to draw over here. This is one of the mitochondria which is spherical in shape, or sometimes it may be a rod shape, and it is one of the double membrane bound structure. It is one of the double membrane bound cell organelle, and this kind of cell organelle has two membranes. So this is called as outer membrane, and this is called as inner membrane. So outer membrane is a smooth one. Outer membrane is a smooth one, and inner membrane has many of the finger-like projections or foldings that we come across. Means here inner membrane is folded and forming finger-like projections, and such kind of projections are called as cristae. Such kind of finger-like projections are called as cristae, right? And there is a space in between. It is called as mitochondrial space. It is about 80 angstroms, right? And these, so here the peculiar structure itself is inner membrane, where it makes finger-like projections, and these finger-like projections are called as cristae. On this cristae, we will find very minute stalked knob-like structures we will find, and this kind of stalked knob-like structures are called as F0, F1 particles, or they are also called as oxysomes, or they are also called as Racker's particles. Racker's particles, they are also called. And so this is more important part of this mitochondria, which is responsible for the production of the energy in form of ATP. So this oxysome or F0, F1 particle or Racker's particle, this particle has three major parts. This is the head part, and this is the stalk part, and this is the basal part. Like this, it has base, it has stalk, and it has head. Such kind of structures that we come across over here. And so all these parts, especially head part, it acts as ATPase enzyme. And this enzyme which help in production of ATPs, adenosine triphosphate. Adenosine triphosphate. So ATPs are produced from these stalked structures, right? They are called as F0, F1 particles or oxysomes or Racker's particles. So now we came to know what are responsible for production of the energy in form of ATPs. Apart from this one, the space in the mitochondria that is filled with matrix. It is filled with matrix, and this matrix itself is slightly jelly-like structure which is present, and that contains a circular DNA over here, a circular DNA, deoxyribose nucleic acid. This is a DNA is present inside it. So this circular DNA, since it is present, these are commonly known as, mitochondria are commonly known as semi-autonomous cell organelles, right? So this is how the structure of mitochondria, right? This is one of the cell organelle which is commonly known as powerhouses of the cell. Okay. Next cell organelle is plastids. Plastids. Let us start. What are these plastids where they are present? Let us go through. Plastids are nothing but these are largest semi-autonomous cell organelles. So why they are called as semi-autonomous? The question arises here. Why? Because these cell organelles, they have their own DNA. They have their own DNA so that they can produce or they can synthesize their own proteins. Hence they are named as semi-autonomous cell organelles. One of the cell organelles is the plastids. And these plastids were first discovered by AFW Skimper. AFW Skimper is the name of the scientist, the one who discovered these plastids for the first time. And these plastids are commonly present in plant cells. And sometimes plastids exceptionally present in one of the animal cells. In the previous video I have mentioned in euglena. Euglena also a kind of plastid is present over here. This is an exceptional one, and in plant cell usually these plastids are present. And these plastids are of three types. There are mainly three types of plastids that we come across. The first one is chloroplast. And the second one is chromoplast, and the third one is leucoplast. Are the three kinds of plastids that we come across in plant cells, right? So first one is chloroplast. It is so familiar to you all. In the previous classes you have learnt about this chloroplast, right? And the next one is chromoplast. So you might know this one also. Chroma means colored. Plastids, these are colored plastids. Leuco, colorless plastids. These are such kind of three plastids that we come across over here. Let us start with one by one. First one is chloroplast. What is this chloroplast? It is the body which contains chlorophyll pigment. Means it is the structure which contains chlorophyll pigment, and it is a self-duplicating one. Why? Because it has own DNA. It can self-duplicate itself, right? Because of the presence of own DNA. Means this chloroplast or the plastids also have their own DNA, right? Self-duplicating large cell organelles in which complete photosynthesis occurs. Such kind of cell organelles are known as chloroplasts, right? So chloroplasts are nothing but they are self-duplicating large cell organelles in which complete photosynthesis occurs. Whenever we go through the structure of this chloroplast, see here, it is also double membraned cell organelle, right? Its shape that differs, its size that varies from one cell to another cell. Especially these chloroplasts are present in the mesophyll tissue of leaf. You might have come across mesophyll tissue of the leaf. Means, so you just go through the structure of leaf, right? This is how the structure of leaf is there, right? When you take the cross section of this leaf, that moment you will come to know, or you will come to this structure. Okay, this is upper epidermis. Upper epidermis. This is lower epidermis, and in between that there is a tissue present inside this leaf, and that tissue is said to be as mesophyll tissue. You are observing here mesophyll tissue. Okay, inside this is completely said to be as mesophyll tissue. Meso means in the middle, phylla means leaf. Middle part of the leaf contains a tissue called as mesophyll tissue, right? In this mesophyll tissue, you will find very minute cell organelles. I am showing only these pointed things. They are nothing but chloroplasts, right? In each and every cell, number of plastids are present, and those plastids, especially chloroplastids, these are chloroplasts present inside. And in mesophyll tissue, we divide it in dicards usually into two parts. One is upper palisade parenchyma and lower spongy parenchyma. Like this we divide. Whatever it may be, in mesophyll tissue the cells are present, and in those cells the dotted things are nothing but chloroplasts, right? So whenever we take one chloroplast, it looks like this. It is a double membrane bound structure. So this is outer membrane, and this is inner membrane. Inside that one, in between that one, there is a space, periplastidial space. Periplastidial space is there, and inside this chloroplast you will find many number of pile of coin-like structures, and those structures which are present inside right? Pile of coin-like structures what are present, a single one. This one is called as grana, singular granum, right? And each coin-like structure is called as thylakoid. Each set of coin-like structures are known as granum. Single one is granum, Yes, plural is grana. Means many granum together form grana, right? So all these grana are interconnected with each other and to the inner membrane, and these are called as fret channels. Fret channels, right? Such kind of a structure it contains. And this granum is major part of this chloroplast. And next one is stroma, the fluid which is fed inside this chloroplast called as stroma. Called as stroma. So in each thylakoid, whenever we go through each thylakoid, they are containing some of the pigments. They contain pigments. Thylakoids, each thylakoid, it is membrane bound structure, flat-eared structure, and each one contains so many of the pigments, and mainly the chlorophyll pigment is present inside that one, and other pigments are also present inside that one, and hence these grana, they help in light reaction. They help in light reaction. And apart from this one, second one is stroma, or it is also called as matrix, right? Matrix, chloroplastidial matrix it is, and this matrix is hyaline in structure. Hyaline means glassy, transparent. So transparent fluid it is. That kind of transparent fluid which is electrode dense. So electrode dense fluid, right, which contains many number of proteins, means it is proteinaceous in nature. It is a highly electrode dense as well as proteinaceous matrix is present inside the chloroplast, which is called as the stroma. So this matrix inside which there a reaction takes place, that reaction is called as dark reaction. We will study this light reaction as well as dark reaction thoroughly in PUC 1 in the chapter photosynthesis in higher plants. Right? In that chapter thoroughly we will study light reaction as well as dark reaction. This is how the structure of chloroplast is, which is present in the mesophils, right? So this is about the chloroplast, which help in photosynthesis. So it is the major function of this plastid, that is chloroplast, is one kind of plastid that we have studied. Second type of plastid is chromoplast. So second one, chromoplast. The name itself suggests chroma, means colored. These are nothing but colored plastids. Plast means plastid. Right? So these are colored plastids. Colored plastids. Means, so these are responsible for the coloration of the flower, fruit as well as leaves. Means these these type of plastids are usually present in fruits, leaf as well as flowers. Means they are present in fruits, leaf and flowers, and these are responsible for color of themselves. They are responsible for the color of the fruit, color of the leaf as well as color of flowers. Right? Such kind of plastids are chromoplast. kind of xanthophyll. Xanthophyll. These are the chromoplast responsible for coloration. And third one is leucoplast. Leucoplast. Leucoplast, as I said initially, these are colorless plastids. What these are? Colorless plastids. There is no coloration they are responsible for. Means, so such kind of plastids are meant especially for storage of oils or fats, proteins, starch, etc. Right? Based on that, there are different types of leucoplasts that we come across. But a very important point to be remembered, it is leucoplasts can convert themselves into chromoplast as well as chloroplast. So they may convert themselves into chromoplast as well as chloroplast. Right? So these are three types of plastids that we come across. Yes. Next type of cell organelle is ribosomes. Ribosomes. These are submicroscopic granular structures present in the cytoplasm of both the plant cell as well as animal cell. Along with that one, they are also present in mitochondria, nucleus, right, as well as chloroplasts, right. Such kind of submicroscopic structures are known as ribosomes, and these ribosomes were first discovered by Porter. Porter is the name of the scientist, the one who discovered this one. And this Porter, he discovered them in animal cell. He discovered these ribosomes in animal cell, and later Robinson and Brown are the scientists seen them in plant cell. And Palade is another scientist, the one who coined the term ribosomes. Coined the term ribosomes for the first time. And how these ribosomes are, already I had mentioned in the previous video, they are bounded on the outer membrane of endoplasmic reticulum, right? I had mentioned this point. And some other ribosomes are freely floating in the cytoplasm. Those are called as cytoplasmic ribosomes. And the ribosomes which are attached to the endoplasmic reticulum are called as bounded ribosomes. That kind of different types of ribosomes that we come across. So types of ribosomes. One is bounded ribosomes, and second one is free ribosomes. Like this we can separate them into two types, right? Another types of ribosomes, already I have mentioned in the previous video, right? These ribosomes may be 70 years in prokaryotes, where they are in prokaryotes they are 70 years, and the second type 80 years type of ribosomes are present in eukaryotes. In eukaryotes we can observe 80 years. What is this S stands for? The question arises here. S is nothing but it is Swedberg's unit. It is Swedberg's unit. What is this Wedberg unit? It is a kind of measurement. It is measurement of the size of the particle, right? So which determines after centrifugation, right? So are determined by the centrifugation process, right? That kind of unit is called as Wedberg's unit, right? Yes, right? In these type of ribosomes, how the structure of ribosomes are. So you can observe here this ribosome structure of a ribosome. This structure of a ribosome has two subunits. It is a smaller subunit and the second one is larger subunit. There are two subunits that we come across, smaller subunit as well as larger subunit. In prokaryotes, these subunits are 30s subunit and this is 50s subunit, right? And in eukaryotes, here also you will find two subunits. One is 60s and this is 40s, right? So these are the two subunits that we come across in ribosomes of prokaryotes as well as eukaryotes, right? There are two types of subunits or two types of ribosomes that we come across. And what chemically these ribosomes are made up of? Chemically, these ribosomes are made up of, one is rRNA, ribosomal RNA we call it as rRNA, along with this one proteins. These two together form the structure of this ribosome. Basically, these two subunits are being separated. This is 40s ribosome in eukaryotes and this is 60s ribosomal subunit. So like this they are being separated usually, but they unite each other during the protein synthesis. Whenever the protein synthesis process that starts, that moment these two unite each other, right? For the union of these two subunits, there is a need of magnesium ion. This magnesium ion is too essential for the union of these two subunits. When both the subunits fuse together and even the RNA, here you can observe here. So this RNA, this type of RNA is mRNA. mRNA that binds to both the subunits and protein synthesis process that continues. Thoroughly we study how the synthesis of protein that take place in PUC too, right? But we came to know these are the cell organelles which are responsible for production of proteins. Hence, these are commonly known as protein factories, right? Ribosomes are commonly known as protein factories. Commonly known as protein factories, right? Next cell organelle we'll see in the next video. Thank you.